Hello, biologists. Today, we are going to talk about dihybrid crosses and non-Mendelian genetics. All right, so you use a regular Punnett square when you have one trait, when we are looking at whether or not the purple flower had the dominant or the recessive trait. We're going to use dihybrid crosses if there's more than one trait, specifically two. If there's three, you can have a trihybrid cross, four quadhybrid cross, but we really stop at dihybrid crosses. So the key to a dihybrid cross is to look at each gene on each the chromosome a little bit separately. So here we have two traits. We have seed color, yellow is dominant, and seed shape, round is dominant. So what would happen if we had a round yellow seed that's homozygous dominant bred with a green wrinkly seed, homozygous recessive? Well, the first step is that you need to figure out what kind of gametes your parents can make. So here we can see up at the top, this is our homozygous dominant plant. And it has two sets of chromosomes. It's diploid. The chromosome 1, which might have the yellow coloration on it, has the dominant allele. And the second chromosome, which has the seed texture, smooth or wrinkly, has the dominant allele as well. When this plant makes haploid gametes, it can put one of each of these chromosomes into its sperm or egg. So if it puts this chromosome into one egg, it'll put this one into another one. And then it could put this dominant chromosome and pair it with this one. The reverse is true. It could take this dominant chromosome and pair it with either one of those other chromosomes. And so you end up with 4 gamete types, but each one looks the same phenotypically and genotypically. Here's another way to think about that. We'll look at the other parent here. Here we have a homozygous dominant, a recessive parent. Here's a visual of the diploid organism's genotype. When this organism produces gametes, it'll put one of these alleles into a sperm or an egg. So here's the little r. And then it's going to pair it with another allele because we need one copy of each of our genes. So it'll be little r, little y. And now it's little r, but we could use the other little y. So we've paired the first little r with both little y's, and we can do the same thing with the second little r. And it does make a difference. So the first little r, now the first little y, because these two recessive alleles that it contains, one is maternal and one is paternal. And so whether or not it passes on its maternal or paternal recessive trait does affect the offspring. So it looks like all of our gametes have the same little r, little y genotype. And for our other parent, the capital R, capital Y genotype. But we know that there's 4 ways to get that, because we can FOIL our different alleles and figure out which paternal and maternal combination there is. All right, let me erase those. That's what I was trying to do there. So that's great. When our organism produces its gametes, let's say this is sperm and this is egg, we're going to have fertilization and we will end up with a diploid offspring. That diploid offspring will have one dominant allele, one recessive allele for each of its chromosomes. So this offspring is heterozygous for both traits because both of its parents were homozygous for dominant and homozygous for recessive. Now, what would happen if we took this individual and we self-pollinate it? And this is how we get to the dihybrid cross. This individual here can also make 4 types of gametes. Everyone can make 4 types of gametes. The first option is big R, big Y. So that's option number 1. That would be a gamete with only dominant alleles. Then we could do big R, little y. that gamete would have a capital dominant trait for color, but not for texture. And then we would do little r, big Y. And finally, little r, little y. So this individual can produce all of those gametes, which means that the type of offspring this individual can have when it self-fertilizes is going to be way more complicated. So now that we've thought about the types of gametes these self-fertilizing individuals can make, we'll then line up those gametes on the dihybrid cross. Here's our big R, big Y, big R, little y, little r, big Y, little y, little r, little y. And we do the same thing for the other parent. So we have the theoretical gametes each of the parents could make, determined by their genotype. And then we drop down and we fill in our dihybrid cross. Now, I like dihybrid crosses. I think they're kind of fun. And when you go through, you can then go and note this one will have the dominant trait for shape, but not the dominant trait for color. And this will have the recessive trait for both shape and color. And if you go through and look at every single trait here, you end up with this ratio. 9 out of 16 will be dominant for both traits. 3 out of 16 will be dominant for one, but not the other. And then 1 out of 16 would be recessive for both. The first rule of probability is when you have two mutually exclusive events occurring simultaneously. That means two events that cannot occur at the same time. For an example, if you roll a dice, you only get one number. So what are the odds of rolling a 1 or a 2? Doesn't happen at the same time, you're wondering about twice. To figure out whether or not you can roll a 1 or a 2, you would take the probability of rolling a 1, which is 1 in 6, because there's 6 sides on a die, plus the probability of rolling a 2, 1 in 6, and you would end up with a third. So the chances of rolling a 1 or a 2 on a die is 33%. Another way we can think about the probability is if it's not mutually exclusive, to independent events that are occurring simultaneously. So now imagine you have 2 dice. So 2 dice, and you want to know what are the chances of rolling a 1 and a 2. What you would do is you would take the odds of rolling a 1, which is 1 in 6, multiplied by the odds of rolling a 2, which would be 1 in 36. Here's another way to practice this idea. Imagine that a couple has 3 children. What are the chances that they will all be girls? Think about the odds of getting a girl, and then whether or not this is independent events occurring simultaneously, or whether or not this is mutually exclusive events. And you would use the rule of multiplication, independent events occurring simultaneously. You would take the chances of getting a girl, which is 1/2, because you're either XX or XY, and then you would times it by the chances of getting a girl, and times it by the chances of getting a girl. So the probability of having 3 girls is 1/8. If a couple has 3 children, what are the probability that all the children will be the same sex? So now we're thinking about 2 things, whether or not they're all girls and whether or not they're all boys. So we'll figure out the probability of getting all girls, which is 1/8, 1/2 times 1/2 times 1/2, and the probability of getting all boys, which is 1/8, and we will add those together. So we've used both probabilities to answer this question. Cool. What if you have a genotype and you don't know if it's heterozygous or homozygous? Then we get to use a test cross. So a test cross is when you take an unknown and you breed it with a known. Now, the known has to be something where you can predict the outcome. Let's have a visual for this. Here's my unknown phenotype for my flower. I could either breed my unknown with a homozygous recessive and have all purple flowers, or if I did it and I ended up with some white flowers, that would tell me something about the genotype of my unknown. So a test cross allows you to look for the recessive allele if it is there. People do this in animal breeding. There are some traits that tend to skip or hide, and so they'll do breeding experiments and see whether or not that gene shows up in the next generation. And because this gene showed up, I know that this purple flower had to have a recessive allele because it got it from both parents. Mendel is having a great time working with his pea plants, but everything that we've talked about so far assumes that there's only one gene that controls the trait, that there's only two alleles, dominant and recessive, and that there's a clear relationship between them as dominant and recessive. Now, that's not the case in real life. Our non-Mendelian genetics include all sorts of really cool things, and we'll go through them one by one. The first thing is incomplete dominance. Incomplete dominance is where your heterozygote is an intermediate between the two homozygous parents. For example, if I had a red flower and a white flower, and it produced a pink flower as an offspring, that's an intermediate between red and white. If I had a black cat and a white cat and they produced gray kittens, that would be incomplete dominance. I could also have co-dominance, where instead of an intermediate, the offspring expresses both phenotypes, but not like their parents. So here I have some camellias. If I took a red camellia and a white camellia, and my offspring was red and white, it's expressing both phenotypes, and so it is co-dominant. We also have polygenic inheritance. Polygenic inheritance is when there's more than one gene. That's what poly means. This is what most of our genes are. Like, most of our genetics are really complex as humans. Example is height and skin color, and they're not totally genetically controlled. There's other factors that affect it, like nutrition affects height. And so we end up with this beautiful spectrum of variation for human height and human skin color, because there's many different genes affecting it and many different other factors controlling it. So this is the most normal type of inheritance for us, is polygenic. We also have multiple alleles. So far we've been writing as a capital or a lowercase, but it turns out we could have several different types. The example that we like is blood type. So blood type is determined by the carbohydrates that you have on your red blood cell. Here's a red blood cell. It's showing you the depression in the center. And then all along the outside, you have these little carbohydrates. If you have A-type carbohydrates, you have A-type blood. If you have B-type carbohydrates, you have B-type blood. And if you have both A and B, then you are AB. And if you have no carbohydrates on your blood at all, you have type O blood. Think O for like 0 carbohydrates. O blood is the most common in America, but it totally changes based on what country you're in. The interesting thing about this is we need to denote our alleles differently. So we think of blood typing as either a dominant or a recessive trait. So we have a capital I or a lowercase I. That capital I refers to having carbohydrates outside of your red blood cell. So you can be capital I, A, and that means that you have A carbohydrates, or you can be capital I, B, and that means that you have B carbohydrates. The recessive is O. So these are the 3 allele types, and each human individual with blood typing is diploid. So if I was type A blood, I would have at least one A, but I could be dominant, homozygous dominant, or I could be heterozygous. That's my little I. And so both of these genotypes give me A blood. It just depends whether or not I also have the allele for O. So I know my blood type, I'm A, and I know my mom is O. So this would be my blood type here, because my dad has A blood, he gave me his A allele, and my mom has O blood, she gave me her O allele. If you have B blood, you could be homozygous dominant for the allele B. Or you could be heterozygous and you could have the allele for O. So in both of these situations, the allele for B or A carbohydrates is dominant to the allele for O. So we have dominance occurring. If you have a B blood, then you're going to have both the A and the B allele. So your blood type would be A, B, and you express both of them. So AB blood typing is an example of co-dominance. Not incomplete, but co-dominance because they're both being expressed. And if you have O blood, the most common type, you have both recessive alleles. And so you have no carbohydrates on your blood type. The really fun practice questions that we could do are things like, if I have AB blood and my partner has O blood, what kind of blood could our child have? And you would fill in a regular Punnett square. So if that's AB blood here, and my partner has O blood, put O right here, then what kind of offspring could we have? We could have offspring with A blood, and we could have offspring with B blood. But we could have no offspring with the same blood type as either of the parents, because my O type blood parent could only pass on the O gene, which is going to get covered up by either the A or the B allele. So 50% of my kids would have A blood, 50% would have B blood. None of them would be the same as either parent. Pretty fun genetic problems here. All right, let's look at our next example of non-Mendelian inheritance. Environmental influence. We talked about Siamese kitties earlier. They have that enzyme that is only functional at low enough temperature, and so it's not functional where the kitty is super warm. And so we have this interesting pattern of colorization. The gene is there, but the gene is being controlled by the environment. And so its expression might not always be visible. Another example is hydrangea. Hydrangeas have the coloration of either like pink or blue, but whether or not it's pink or blue depends on the pH of the soil. So acidic soil and basic soil create different flower color. The phenotype changes with the soil pH. And that means I have a blue hydrangea out back. I could change it to a pink one by altering the pH of the soil, and then I could change it back. So you could swap it. This is one plant here where half of the plant is exposed to basic or acidic, and the other half is exposed to the other one. And so the environment is altering the phenotypic expression of the genes. And then the last one that we can discuss for non-Mendelian inheritance is sex-linked traits. Sex-linked traits are on the X chromosome. There's really not space for anything on the Y chromosome, so we often refer to sex-linked traits as X-linked traits. So both those vocab words are used. We have a lot of examples. Some are dominant, some are recessive. Recessive is much more common. And the way that this looks when we do a Punnett square is that we put the trait on the X only. By the way, this is a color blindness test. If you look at this circle and you cannot see the number in it, you may be red-green colorblind. Please don't let this diagnose you. Go talk to your doctor. But color blindness is often a sex-linked trait, and so it's cool to think about. So here's what it looks like. If you have a X chromosome, then you will have the trait on it. Let's just do A's. So this person is homozygous dominant and a female. This person is heterozygous and a female. And this person is homozygous recessive and a female. Remember that sex and gender are different, so we're talking about chromosomal sex. Now let's look at males. If we have a male with these traits, I'm going to divide them. I could have a male that has the dominant allele, but males have Y chromosomes, so they only have one X chromosome, and the Y chromosome does not get the allele. The allele is only on the X chromosome. So males have 2 options, where females have 3 options. That's a little a. And that means that the chances of expressing a recessive X-linked trait for a male are 50%. Because there's 2 chances right there. The chances for expressing it as a female, if it's a recessive trait, is only 33%. Because there's an option of being heterozygous. And if it's a recessive trait, that dominant allele will cover the recessive allele and you won't express the phenotype. So this explains why males are more often to be colorblind, more often to have types of muscular dystrophy or hemophilia, and it's just because it's on the X chromosome only. Let's check out what a Punnett square looks like with a sex-linked trait. Here's the Punnett square. Now in this Punnett square, they put it as a subscript instead of a superscript. It doesn't matter. What's important to note is that there isn't anything on the Y allele, or on the Y gene, that it's only on the X. So when this individual here, who is a dominant presenting male and a recessive presenting female, if they have children, they're going to end up with a carrier daughter because she has the allele, but she is not expressing it. That dominant allele is hiding the fact that she could be colorblind. A second carrier daughter who is not expressing colorblindness. Where did these dominant alleles come from? They came from dad. Even though mom is colorblind, none of your daughters will be colorblind because dad's X is protecting them from colorblindness. Now, of the sons, we're going to have 2 colorblind sons because both of them have the recessive allele and they don't have a second X chromosome to hide it. So all of your sons will be 100% colorblind and 0% of your daughters will be colorblind. It's important to note a carrier is someone who is heterozygous for that trait, and the affected person is going to be the recessive allele bearing person. Okay, so I hope that was fun, and email me with questions. We will talk soon.